Welcome everyone, I'm Shafiq Rahman, and today with this presentation I'm sharing findings of our recently completed project under the title Antimicrobial Resistance Surveillance from Farm Aquatic Foods in Wet Markets and Antimicrobial Use Prevalence in Bangladesh Aquaculture. Antimicrobial resistance, or in short, AMR, is a globally recognized health problem, posing major threat to public health management of infectious diseases. On the other hand, aquaculture, one of the leading food sector, going through huge transformation towards intensification, which is linked with the increasing outbreak of infectious diseases in aquaculture farm, leading to irrational and inappropriate use of antibiotics. The industry is considered one of the hotspots of emergence and the persistence of AMR pathogens. Resistance pathogens can easily spread through interconnected agri-food system. In Bangladesh, use of antibiotics in livestock sector is unknown. However, information in context to the presence of AMR pathogens at point of consumption and the factors driving use of antibiotics in aquaculture in Bangladesh is very limited. This particular research is mainly focused on two objectives: generate evidence of antimicrobial resistance in key pathogens from farmed aquatic foods and the characterize antimicrobial use in commercial aquaculture in Bangladesh. We have collected 132 samples of shrimp and tilapia from 2 wet markets. Bacteriological samples are taken from skin, gill, muscles, and the intestine, then enriched and cultured on selective media for primary identification. 4 bacteriological isolates, Escherichia coli, Salmonella, Vibrio parahemolyticus, and the Streptococcus were considered for this study. Positive isolates were PCR confirmed. This detection method was used to check the susceptibility of the antibiotics. Results are interpreted using CLSI breakpoints. In parallel, we also conducted point prevalence survey in 6 districts covering a total of 672 aquaculture farms. Our AST results indicates that both E. coli and the Salmonella were major resistance to ampicillin. In addition, all Vibrio parahemolyticus showed resistance to ampicillin because of their intrinsic resistance to the antibiotic. Streptococcus isolates are resistant to azithromycin and nalidixic acid, indicating possible multiple drug resistance. The point prevalence survey we conducted identified 7 antibiotics commonly used in aquaculture farm in Bangladesh. Among these, oxytetracycline was identified one of the most commonly used antibiotics. Fish farmers uses antibiotics both for prophylactic and therapeutic purpose. Our study reveals that a number of socioeconomic factors are associated with antimicrobial use in aquaculture farm in Bangladesh. Farming practice, occurrence of illness in fish, aquaculture training, poor knowledge on antibiotic uses, and shorter farming experience were significantly linked to the use of antibiotics in commercial aquaculture farms. In conclusion, the research work confirmed the presence of antimicrobial resistance bacteria in fish and shrimp samples. Initial whole genome sequencing confirmed the predicted antimicrobial resistant genes. Point prevalence survey results coincide with the previous observation of uses of antibiotics in aquaculture in Bangladesh. We are good at factors associated with antimicrobial use in Bangladesh aquaculture are also embedded in aquaculture practice and socioeconomic heterogeneity. Thank you very much for your attention.